Look at a dividing cell. It builds intricate structures and replicates DNA with absolute precision. Contrast that with an abandoned building slowly crumbling into dust. That decay is driven by the second law of thermodynamics. The physical universe operates with a strict tendency to move from order to maximum chaos. Things naturally fall apart. They do not spontaneously assemble themselves. If the laws of physics dictate that everything must break down over time, how does a highly ordered living organism manage to exist, thrive, and replicate? Biological systems operate as highly efficient thermodynamic engines. Every process inside a living cell is governed by a strict energetic ledger, known as bioenergetics. Bioenergetics is the quantitative study of energy relationships and conversions in biological systems. Whether a cell is synthesizing a protein sequence or contracting a muscle fiber, every action requires a payment. It is a transaction within a tightly regulated chemical economy. This cellular economy is dictated by three specific variables: enthalpy, entropy, and Gibbs Free Energy. Mastering the relationship between these three variables is the key to understanding how biological work is achieved. We start with enthalpy, represented by H. Enthalpy is the heat content of a system, representing the potential energy stored within molecular chemical bonds. Molecules do not fall apart on their own. Breaking an existing chemical bond requires an input of energy from the surrounding environment. When those atoms rearrange and form new bonds, energy is released back out. The net difference in this exchange is the change in enthalpy, or delta H. If a reaction absorbs more heat than it releases, it is endothermic. If it releases more heat than it takes in, it is exothermic. Cells must constantly manage these heat exchanges to fuel physical changes at the molecular level. Heat is only half the equation. We also have to account for entropy, or S. Entropy is the strict quantitative measure of randomness, disorder, or freedom of movement within a given system. This illustration compares an ordered brick wall to a disordered pile of bricks. Dropping the bricks creates the disordered pile, which is a highly probable state representing a positive change in entropy. Entropy increases as movement becomes less constrained. Molecules in a solid state are tightly packed with very little freedom. As they transition to a liquid and then to a gas, their freedom of movement increases. This creates a biological paradox. A cell maintains itself by creating intense local order, decreasing its own internal entropy. To do this while obeying physics, the cell releases heat into its environment, increasing the total entropy of the surrounding universe. Entropy acts as an unavoidable thermodynamic tax applied to every biological action. To determine if a cellular process can occur, we combine enthalpy and entropy into a master equation. Delta G equals delta H minus temperature times delta S. The result, Gibbs Free Energy, or G, represents the total amount of energy available to do biological work. A negative delta G indicates an exergonic reaction. These are spontaneous and proceed on their own without requiring extra energy. A positive delta G indicates an endergonic reaction. These are non-spontaneous and absolutely require an external energy input to occur. Gibbs Free Energy is the ultimate chemical threshold. If a biological process has a positive delta G, it is physically impossible for it to occur on its own. To pay for these impossible reactions, the cell relies on ATP. The hydrolysis of ATP carries a highly spontaneous free energy change of negative 30.5 kilojoules per mole. This image shows the chemical structure of ATP. Note the cluster of negatively charged oxygen atoms packed together along the phosphate tail. Because like charges repel, these four negative charges are violently pushing against one another, creating intense electrostatic repulsion. When water is introduced during hydrolysis, it cleaves off the terminal phosphate group, instantly relieving that electrostatic tension. The newly freed inorganic phosphate is immediately stabilized by taking on resonance hybrid structures, spreading its electrons into a more stable configuration. Both the resulting ADP and the free phosphate are heavily solvated by surrounding water molecules, pushing the system toward a stable, lower energy state. The utility of ATP lies in its thermodynamic instability. It is a tightly coiled molecular spring, existing specifically to release its tension. The cell faces a persistent problem. Many critical survival reactions, like the initial phosphorylation of a glucose molecule, are endergonic. By themselves, they are simply thermodynamically impossible. Cells use energy coupling, linking a non-spontaneous reaction to ATP hydrolysis. Glucose phosphorylation requires positive 13.8 kilojoules per mole. ATP yields negative 30.5. Because these equations are additive, combining them results in a spontaneous net negative 16.7 kilojoules per mole. By mathematically merging these chemical events, biology bypasses the thermodynamic barrier, forcing life-sustaining reactions forward. This exact thermodynamic accounting dictates biological survival on a macro scale, a scale we can actually see with the naked eye. That half-second snapping movement relies on an instantaneous change in turgor pressure within the plant's mesophyll cells. To generate that pressure shift, the cells aggressively pump potassium ions across their membranes against their natural concentration gradient. Moving ions against a gradient is an endergonic process. It is driven by coupling the movement to the negative 30.5 kilojoule release of ATP hydrolysis. The principles of enthalpy, entropy, and Gibbs Free Energy are the physical instructions powering every movement, reaction, and function of a living organism.